Good morning, all, and welcome to the Center for Iron and Heme Disorders Virtual Seminar Series. Today, we are lucky enough to have Dr. Merav Socolovsky speak to us, and the title of her talk today is The Role of the Cell Cycle in Erythroid Differentiation. Merav got her bachelor's degree at Cambridge in medical sciences and also an MBBS degree in medicine and surgery. She then got a Ph.D. at the University of Cambridge in physiology, and I noticed she did a postdoc in Harvey Lodish's lab, working at the Whitehead Institute on EPOR signaling. There's a lot of accolades I could list, but I really want to hear Merav's talk, and so we are going to turn the time over to her at this point. And thank you so much for speaking.
It's an absolute pleasure. Thank you so much for inviting me. And I'm delighted for this opportunity to talk about what we've been working on in my lab, which is the connection between the cell cycle and erythroid development. We've been working on this for 20 years now. Really, we haven't started out trying to work or wanting to work on the cell cycle. It's just that while working on erythroid development, the cell cycle kept showing up, and I'll show you how. So first of all, can you hear me okay?
Yes.
Yeah, perfect. Okay, so just trying to, yeah. So fate decisions. So this is a fundamental question in biology. Many labs working on this question in many different models. And erythropoiesis turns out to be an interesting model for that. So we have in erythropoiesis, early erythropoiesis, where hematopoietic stem cells give rise to multipotent and then erythroid progenitors, which undergo self-renewal and amplification. They're not morphologically identifiable, but then at some point they enter terminal differentiation, and at this point they become morphologically identifiable erythroblasts. And so the question that we asked is, how does that transition happen? And there are, I'm having trouble transitioning the slides, sorry. Not sure why. So the question is, how does that happen? And so there are perhaps two diametrically opposed ways in which we can think of it. Maybe there are incremental changes that gradually transition progenitors to erythroblasts, but perhaps it's a switch. And what do we mean by a switch? Switches in biology suggest some kind of toggle switch, or it's also known as bistability, meaning that we have two relatively stable states and an intervening bistable state. And these situations happen when the two states that toggle are incompatible. You can't possibly have a cell being in both states. And we know such situations. For example, a cell can't both self-renew and differentiate. You do either one or the other. You can't be both quiescent and proliferating. Or survival state is another one. A cell is either viable or has entered a programmed cell death. So that's in a very specific way, and usually contains in the wiring diagram, if you like, what's called a positive loop. And one such positive loop is the double negative loop. So this type of loop will lead to positive feedback. So if we see a slight change in one direction, it will feed on itself, and the state will switch from one to another. And I think this is pretty obvious, but there might be a small differentiation signal that will push this one regulator up, which in turn will push the other one down. And if that gets started, it will rapidly progress. Okay, so the model that we've started studying is the mouse fetal liver. And this is pretty familiar now, I think. with CD71 and tau119, we can divide it into 6 subsets. And we know that all of the early progenitors are in subset 0 and 1. So, and that's because that's where we see colonies, mainly CFUE colonies. And once we've moved into subset 2, that is, once we see tau119 being expressed, that's a terminal differentiation gene. That means we're already in terminal differentiation. The cell no longer make colonies, and they are progressing in the differentiation pathway, internal differentiation. So early on, we realized that there is a clear divide between CFUE cells in S0, S1. Both of them make CFUE colonies, but the type of progenitor that give rise to these colonies is different. And the first hint came from studying the EPO receptor knockout embryo. So when we take the fetal livers of each of these littermates and look at them using flow cytometry, we see that this is around embryonic day 12.5. This fetal liver already has erythroblasts in S3. That's basophilic erythroblasts. But in the knockout, we have an absolute block at the transition from S0 to S1. So S1 CFUE cells are dependent on EPO. The second thing that we realized is that something interesting is happening with the cell cycle. So this is the assay that we use a lot. It's the BRDU pulse assay. What you do is take a BRDU, which is a thymidine analog, pulse. You could do it in vitro, but we've done it in vivo. You inject the pregnant female mice with BRDU, and then 30 minutes later, you harvest the embryos and the fetal livers. And that allows you to determine by FACS the cell cycle status of each individual cell. So based on DNA content and BRDU incorporation. So all the cells that incorporated BRDU must have been in S phase at that time during the 30-minute pulse. And then based on DNA content, we know where in S phase and whether the ones that are BRDU negative, whether they're in G1 or G2M. And so this assay also tells us one more thing. That is how much BRDU is incorporated during the 30-minute pulse. So if we see cells that have been subjected to the exact same pulse in vivo, but have incorporated different levels of BRDU, that tells us that the rate of DNA synthesis is very different. Incorporated BRDU more slowly must have a longer S phase compared with this cell type. Okay, so with this introduction to the assay, this is what we found when we looked at the fetal liver. So looking at S0 cells, we have cells in S phase.
Merav, you're periodically freezing. I'm not sure why.
That the cells are essentially synchronized in S phase. This is one explanation for why they're nearly all in S phase. They're nearly all synchronized in S phase at the time that they transition from S0 to S1. There's another reason why they're nearly all in S phase, which is that the cycle is structurally different. There is a very short S phase, but the cycle itself is extremely short, and gap phases are very short. So S phase dominates nearly the whole of the cycle. Okay, so the reason why they're nearly all in S phase, we've just covered briefly. It also turns out that S phase speed is 50% faster in S1 compared with S0. And you could see this. There's a lot more BRDU on average incorporating into these cells than into S0, or indeed into cells subsequent to S1. So the speed of S phase peaks in S1. And so to summarize a lot of experiments that we did a while ago, what we now know is that the transition from S0 to S1, and remember these are flow cytometric subsets, so we don't yet know exactly how they correspond to functional developmental stages in erythropoiesis. But in terms of flow cytometry, going from S0 to S1 happens during S phase. It's an S phase dependent transcriptional switch. And that S phase is both faster and shorter. Okay. And it coincides with an epigenetic switch. So when we look at chromatin, we see changes. We see changes in chromatin accessibility at the beta globin locus control region, for example. We see changes in histone tail modification. It's at this point that we see cells entering a state of global demethylation. So starting at the transition, they start to lose methylation genome-wide, losing about a third of all genome methylation during terminal differentiation. And we also see changes in the 3D structure of chromatin. It's at this point that we see the onset of erythroid gene induction, the kind of erythroid genes that we know are relevant to terminal differentiation, like ALAS2 and BAN3. And it is at this point that we see the onset of hypodependence. So all of these changes, to the extent that we could test their dependency on the cycle, all dependent on entry into S phase and transitioning from S0 to S1. Okay, so what might be underlying this? We don't really know yet entirely, but part of the story seems to be that it is at this point that we're losing PU1 protein from the cell. So PU1 declines rapidly at the transition from S0 to S1, and that decline depends on entry into S phase. And it's a double negative interaction. So as soon as the cells enter S phase, P1 begins to decline. But as soon as P1 begins to decline, the cells are allowed to enter into S phase. So if we maintain P1 at high levels, the cells fail to enter S phase. So this is a classic double negative loop. And it may be what underlies the bistability of this transition. So we have a switch-like transition into S phase and into terminal differentiation. Okay, so I mentioned that S0 and S1 are flow cytometric subsets. And one possibility is that because S0 contains multiple developmental stages, going from hematopoietic stem cells to multipotent stem cells and so on, it's possible that what looks like a switch, in fact, isn't, that it hides multiple incremental stages that the cells undergo internally, and it looks like a switch for the artifactual reason that we're looking at a pooled set of developmental states. So to look at that, we had to look at single cells. So that's something that we were able to do in collaboration with the Klein lab a number of years ago. And so we undertook single cell RNA sequencing. Here we're looking at the UMAP projections of the fetal liver and the bone marrow. Topologically, they're very similar. What we see is that the erythroid branch, which starts with multipotential progenitors and extend in this direction, is massive in the fetal liver, but it's still pretty big in the bone marrow. And it has this bulge. Initially, we called it the bulge. And really what this tells us is that we have many cells that are transcriptionally similar. So they're all sort of clustering in the same spot on this UMAP plot. So that means that we have some level of self-renewal here, some amplification. And the same happens here in the fetal liver. So we focused on the erythroid branch. The red color tells us the probability that a cell will progress into the erythroid fate. And we, just to show you quickly how a number of classical genes look, so CD34 is high in multipotential progenitors and then declines rapidly as cells enter the erythroid branch. This is GATA1 and alpha globin. So we developed a flow cytometric method that we've been using really ever since. And this method allows us to identify cells at different locations within this plot. So we can identify cells early on, cells in the branch that leads onto this bulge, and then cells in the bulge itself. And we gave them very imaginative names, P1 for population 1, P2 for population 2, and so on. But functionally, we then went on to analyze what each of these populations does using a number of different assays. And this is a summary that gives us three different ways of identifying each of these populations. So we have the EBMP cells, which stands for erythroid basal mast cells and megakaryocytic progenitors. So these are cells that early on can go down each of these three branches, so megakaryocytic, basophil mast cells, and erythroid. And they correspond to P5 in our FACS strategy. Then we have these cells in this narrow neck. That means we have rapid transcriptional change when cells are here. This stands for early erythroid progenitors. Functionally, they turn out to be BFUE. And they're very purely BFUE, and they contain essentially all of the BFUEs in the bone marrow. So this is P2. And then P1, which is almost entirely CFUE, both early and late, and we divide them into early and late CFUE based on CD71 expression. And there's a small, if you like, contamination of BFUE here as well. Okay, so with this tool, we were now ready to look in more depth into what happens at the transition from early erythropoiesis to terminal differentiation. So here is our cartoon. That's the transition. And here we're looking at all the genes that are changing during the transition from hematopoietic stem cells all the way to mature red cell. And they are organized in order of their peak expression. And so what you can see is that we have a number of key stages, and that's sort of visible. We don't have to do any clever analysis to see this. And really, most important is that we have a group of genes that are expressed really for quite a substantial period in terms of the number of cell cycles. So this corresponds to the CFUE stage. So the CFUE stage expresses, if you like, a dedicated program for that type of progenitor, and it is relatively stable, as you might expect for a cell state that is mainly undergoing amplification. And then at some point, most of these genes turned off, and a whole set of new genes turns on. And that's the change from the CFUE cell state to terminal differentiation. So looking at the single cell level at what happens transcriptionally, the transition from CFUE to terminal differentiation to ETD is a transcriptional switch. So if it is a switch and we now have single cell data, we can ask what is the context of that switch? What might lead to it, up to it? And the first thing that we note is that looking at the expression of CD71, it goes up gradually and then takes off rapidly. And that's what we see as we transition in the fetal liver from S0 to S1. So the upregulation of CD71 really does mark the transcriptional switch. So it's a great marker for the transcriptional switch that we're looking at. So that confirms what we've known functionally. When we look at transcription factors, and I won't go through them, many of them are changing, but very slowly, and none of them are changing impressively at the time of the transcriptional switch. So transcription factor expression by itself doesn't seem to be sufficient to explain the reason for the transcriptional switch. When we look at the cell cycle, however, and here we're looking at cell cycle phase, so genes that correspond to either G1, S phase, and so on, are all plotted together. What we see is that throughout differentiation, we don't really see very much. So that means that cells are in they're asynchronous in terms of the cell cycle phase that they're in at any one point in time here. But when we get to the switch, we see the formation of a number of peaks, starting with the red and the orange peaks, so G1 and S phase. So the switch corresponds to G1 and S phase. So based on single cell transcriptional state, we can see that S phase is absolutely synchronized with the onset of the switch from CFUE to terminal differentiation. Okay, so the next thing that we asked is, okay, so what might be leading up to the switch? And so to look at that, what we did is we looked at this bunch of genes that are changing and asked, which of these genes is most highly correlated with progression as we transition through, with progression along this pseudotime? And the genes that we got back, the Go groups, were almost entirely cell cycle related. So DNA replication, metabolic processes related to DNA, and so on. And here are some examples. And you could see that there is a very tight correlation between progression along X and progression along Y. And all of these are cell cycle related genes, starting with E2F4, cyclin E1, cyclin A2, both peaking at the switch. These are S phase cyclins. Proteins that play a part at the origin of replication. Cyclin D, however, is highly anti-correlated, as is cyclin CDK6. These are G1 cyclins and CDKs. Okay, so how do we explain this? Why do we see this pattern? And so we did a functional cell cycle analysis of the type I showed you earlier in the fetal liver. Here we're looking at the bone marrow. We see the same in the fetal liver. And we can isolate these cells now using our FACS approach that I've shown you earlier. So we can look at BFUE and CFUE. And we've looked at very slim incremental steps progressing through from BFUE through CFUE using CD71 gates. So we know CD71 increases gradually throughout differentiation and then very rapidly at the transition. So we made use of that. And what we see is that as we progress gradually from BFUE into CFUE and through the CFUE stage, the number of cells in S phase increases gradually, and the number of cells in G1 decreases. And then looking at the speed of the cycle, we have some change here, but relatively speaking, S phase speed is constant until we get to terminal differentiation where it increases. So that tells us the length of S's is more or less constant. And the reason for the decreasing in G1 and the increasing in S phase can only be explained by suggesting that G1 phase is gradually shortening as we progress through early erythropoiesis. And that would also have to mean that the cell cycle itself is undergoing gradual shortening. Not because of S phase, but because of G1. So just to recap, what we know so far is that we have a gradual shortening of the cycle as a result of a gradual shortening in G1. And I don't show you the data today, but we know now through an independent method using a mouse that is transgenic for a cell cycle length reporter that was made by Shanshin Guo in Yale, that this is correct. So we do see the cycle undergoing gradual shortening as we transition through these early phases. And then at the time that the cells transition into terminal differentiation, at this point, we see a further shortening of the cycle, but this now is the reason, is caused by a shortening in S phase itself. And then as the cells progress into terminal differentiation, they do undergo a number of short cycles, but once they progress further into terminal differentiation and towards the end of differentiation, the cycle lengthens again. Okay, So to summarize what I've shown you so far, um, we have a rapid S-phase dependent switch that takes cells from CFUE to the ETD phase. The switch coincides with a very fast S phase, and there are structural changes in the cycle leading up to the switch and at the switch. Now, one reason for the very fast cycle and fast S phase at the switch is DNA demethylation. This is work that we did a number of years ago, and I won't go into it, but it does depend on the very fast S phase, and it does facilitate transcription of erythroid genes of the terminal differentiation program. Okay, so is fast S phase important? And I'm kind of running out of time, so I won't go into any details, but there are a number of known cell fate transitions that do seem to coincide with particularly short S phases. And so it is worth asking, what is the mechanism that underlies the short S phase, and why is short S phase important? And we don't really know either of these things fully for any of these systems. And I actually think that we are most advanced in erythropoiesis. Okay, so there are two different ways in which to make SL go faster in S phase. One is to have more origins. And so if the replication starts at more origins, it will be completed sooner. And the other is to have the same number of origins, but for the forks to progress faster. And of course, you can have both. And so we asked which is it a number of years ago, and we used a technique called DNA combing, where you isolate the genomic DNA and then you stretch it on a slide. And before doing this, you label the cells with thymidine analogs. And what we did is took fetal livers, labeled them with one analog, then with a second analog, and then asked how far did replication forks progress. And this is the kind of picture that we get. Blue stains the whole DNA fiber, and then it's easier to look at red and green here. So green is where we start, so that's where the origin will be, right in the center. And then two forks progress in opposite directions, and then the red gives us the direction of each fork. So with this approach, we measured both the number of origins and the speed of the forks, and what we found is that it's the forks that are moving faster. The origins, in terms of their frequency, don't really change as you go from S0 to S1. It does change later on in terminal differentiation. Okay, so we have the forks moving faster. This is a global change. We don't actually know which forks at which genomic location move the fastest, and that's something we're interested in doing right now. The technology wasn't there, but it's now come on board, so we might be able to deduce that. So we have globally faster forks, and that's the reason for the shorter S phase. And so what might be regulating that? So in the remaining parts of my talk, I will look at that question. And we have two regulators. The first one is P57. These are two regulators that we've analyzed so far. There are probably many others. Okay, so CDK inhibitors, P57 is a CDK2 inhibitor. It also inhibits other CDKs, but in the erythroid system, it seems to be specific for CDK2. It's expressed particularly in S phase of S0 cells, and it declines rapidly as the cells transition to S1. And what it does in S0, in S phase, sorry, is to slow down replication forks. So we looked at the P57 knockout. We found that these embryos were anemic. They had smaller fetal livers. Differentiation wasn't as good as it was in wild-type littermates. And the reason for that turns out to be that they fail to undergo efficient self-renewal of CFUE. So the cells don't self-renew well, and they die instead of undergoing switching to terminal differentiation. And what we saw when we looked at the cell cycle was that CFUE in the p57 deficient embryos had even in S0, so they had a prematurely fast S phase. When we did DNA combing, here we're looking at the fetal liver S0 and S1 in the p57 knockout fetal livers. What you can see is that their forks are almost as fast as the S1 forks of the wild type. And then they go even faster in S1. And it seems that this very fast S phase somehow interferes with self-renewal of CFUE. We took those CFUE, that is S0 cells from the fetal liver of the p57 knockout and wild type littermates and tried to amplify them in the dish. And a very well-known method of doing that is to put these cells in the presence of glucocorticoids. That preserves the CFUE cell state and allows the cells to undergo a number of amplication self-renewal divisions in the dish. So when we did that, the wild type cells underwent self-renewal, self-divisions for a number of days before differentiating, but the p57 deficient ones weren't so good at doing that. So what we did next is say, well, is it just S phase speed? And so to look at that, we said, well, what p57 does is inhibit CDK2. There are many drugs that will do that. So we took the p57 deficient fetal livers and in the dish added CDK2 inhibitors. We used roscovitin, we used other inhibitors, and this is what we got. So here is the knockout. It undergoes very poor self-renewal in the dish. So these are the CFUE cells. CFUE cells of the wild type underwent very prominent self-renewal, many cell divisions before differentiating. And the knockout with the drug, so when we inhibit CDK2, that is when we prolong their S phase, they regain the ability to self-renew. So this is our model. What we think is happening is that the long S phase of early erythropoiesis is in fact necessary to maintain the stability of the transcriptional state of CFUE cells. If S phase becomes short, the CFUE cell state is now unstable, and they're more likely to switch to terminal differentiation. And what happens in the p57 knockout is that happens very early on. Okay, so what happens in CFUE cells that are growing in the presence of glucocorticoids? It turns out that what the glucocorticoids do, the reason they are promoting self-renewal of CFUE in wild-type CFUE, is that they in fact induce p57. So in the presence of EPO, CFUE will differentiate and p57 declines. It's actually this lower band. p57 is there weakly and it goes away very rapidly. But if we add glucocorticoids like dexamethasone, p57 is induced and is maintained at high levels. And it is this that allows the cells to maintain a long S phase and to maintain the CFUE cell state for more cell divisions. And so what we ask next is, can we do this in vivo? And this is something we're still working on, but these are some of our results. So what we did is inject mice with a CDK2 inhibitor, and we do find in vivo that the number of CFUE increases, as do all the CFUE progeny. So we see higher levels of proerythroblasts, erythroblasts, and even reticulocytes. And this is only after 48 hours. So we are investigating this further. Okay. So in the last few minutes, I will talk about one other regulator of the very fast S phase at the switch. So just to recap, If we have hypoxia or anemia, that will induce glucocorticoids, but it will also induce EPO. What glucocorticoids do is through induction of P57, and glucocorticoids do many other things, as many other labs have shown. But what we have found is that P57 is essential to their function. So when P57 is induced, we see a longer S phase maintained at the CFUE stage that promotes CFUE self-renewal, and that will expand the CFUE pool and contribute to higher rates of erythropoiesis. At the same time, we see EPO go up. And the question is, what does EPO do? Well, it's very well known that EPO promotes the survival of late CFUE and early erythroblasts. But is that all it does? So whether EPO also affects the cell cycle was controversial, and for a long time, people believed it doesn't. And the reason that they believe that is that they looked at the fraction of cells that are in each cell phase, so the fraction of cells in S phase, the fraction of cells in G1. And it turns out that it really doesn't change that. But it does change the number of cell cycles, and it does change the speed of those cycles. So we have shorter cycles and more cycles. And I will show you the data now. Hopefully, I'll be able to do that in 5 minutes. Okay, so this is work that was done by my most recent PhD student, Daniel Hidalgo, who's now graduated. And what he found is that in the presence of high levels of EPO, early erythroblasts divide more times and divide faster compared to low levels of EPO. And they're also bigger. And that's kind of a puzzle. Okay, so the system that he started with was looking at the EPO receptor knockout mouse. And then those fetal livers, those cells clearly die. So to stop that from happening, we introduced BCLX into those fetal liver cells and compared them with cells from the same EPO receptor knockout where we introduced the EPO receptor back. And then we compared these two cell states. And so when we looked at these using cytospins, we could see, obviously, in the absence of EPO receptor, we see no differentiation. In the presence of EPO, when we put EPO back again, EPO receptor back again, we see differentiation. The brown coloring is diaminobenzidine, so this is hemoglobin being expressed. We see an erythroblastic island. And then in the presence of BCLX, the cells also differentiated, but they did it faster. And they also made much smaller cells, as you could see. So we did this quantitatively. We looked at the colonies that these cells were making. And in the presence of BCLX only, we had much smaller colonies. And so we asked, why is that? Are the cells smaller, or are they dividing fewer times? It turns out both are true. And so here we're looking at the cell cycle. So when we put EPO receptor back, we see S phase in these cells. We see the cells also entering S phase when there's only BCLX put back. But when we compare the rate of BRDU incorporation, you can clearly see that in the presence of the EPO receptor, S phase is much faster than in its absence. And that is true. Here we're looking at S phase speed throughout ensuing differentiation, and you can see that it's consistently faster. The number of cells in S phase is also higher. And we see that the number of cell divisions is higher. So here we're looking at a number of different ways that we were trying to rescue the situation. But the bottom line is cell viability is about the same when we put BCLX or EPO receptor back, but the number of cell divisions is faster if we put EPO back compared with BCLX. And the cycle is shorter for EPO than it is for BCLX. The same is true in vivo. So this is a mouse that will tell us how fast the cycle is in vivo. And it turns out that this is a report. I won't get into it, but it basically tells us cell cycle length is shorter when we inject mice with EPO compared with mice that are injected with saline. And Janice is on the call. I remember showing these results to Janice well before they were published, and she said, well, maybe BCLX is overexpressed. Well, no, it's we're definitely expressing it, but maybe it is overexpressed, and maybe it is BCLX overexpression that makes the cells divide fewer times or be smaller. So that was very clearly a very possible scenario. So to look at that, what we did is infected cells either with EPO receptor alone, BCLX alone, or both EPO receptor and BCLX, and they're on different colors, so we could tell them apart. And now we looked at cell size, and here we're looking at cell size using imaging flow cytometry, and we're looking at reticulocyte cell size, but this was true for all erythroblasts too. So we could see that BCLX alone, we have smaller reticulocytes than in the presence of BCLX and EPO receptor. So BCLX overexpression by itself wasn't sufficient to do that. So what we think is happening is that we need EPO receptor signaling to have larger cells and faster cycles. So I'm running out of time. I'm going to skip this. This is just to show that this is nothing to do with iron metabolism. And it does happen in a way that is responsive to EPO concentration. So obviously EPO concentration, this is an old paper that is a classic telling us how EPO levels rise as anemia becomes more and more severe. And what we did here is take mice, inject them with increasing doses of EPO, and looked at erythroblasts at different stages in a way that is cell size independent. And I won't go into it, but we effectively use nuclear offset. Obviously, if we're looking at cell size, we can't use that as a measure of differentiation stage. And you could see that at each stage, the higher the EPO was, the bigger the diameter. So we have, to summarize, before I get to summary, the same is true in human volunteers. So we collaborated with a number of European groups who were giving healthy volunteers EPO. This is to study doping. And it turns out MCV was higher and persisted as a high even after reticulocytes came down. So we have a high MCV when EPO levels are higher. So we have here a paradox. EPO does two things. So we know that during terminal differentiation, the more cell divisions we have, the smaller the red cells that are being made, and vice versa in vitamin B12 deficiency. And so we expect that if EPO receptor signaling induces more cell cycles, we should get smaller erythroblasts and smaller reticulocytes, but that's not what we see. We see the opposite. We also know that if we have a longer cycle, the cells have more time to grow, usually in G1, and so we will have larger cells after cell division compared with cells that undergo division after a shorter cycle. So we expect, again, EPO receptor signaling induces shorter cycles. We should see smaller cells. And yet we see that in the presence of EPO receptor signaling, the cells that we get at every stage of terminal differentiation and the reticulocytes that we get are larger. So the only way to solve that problem is to hypothesize that EPO receptor signaling also promotes faster growth. In other words, faster protein synthesis, faster ribosome biogenesis, and so on. And these are processes that we are investigating right now. Okay, so to summarize this last part of the talk, EPO receptor signaling increases both cell cycle number and cell cycle speed, and paradoxically, also cell size. This is not to do with alterations in iron metabolism, which we've shown genetically. And we think that it suggests that EPO receptor signaling promotes exceptionally fast growth in biomass. And we are trying to understand those processes right now. And it also suggests that there might be new interpretations of high MCV that all other things being equal, for example, no nutrient deficiencies, MCV might indicate the level of aporeceptor signaling and perhaps hypoxia. So I'd like to just thank people in my lab and some of my collaborators. So especially I'd like to point out a long client with whom we've collaborated on single-cell RNA sequencing, and we're still collaborating on other projects. And Nikolai Nordsburg in Europe, who gave us access to his data on healthy human volunteers, and people in my lab who did the work, particularly Daniel Hidalgo and Yang Huang. So thank you. And if there is time for questions, hopefully I'd like to take them.
Thank you, Merav. That was wonderful. I want to invite people to go ahead and ask questions at this point. We have about 5-10 minutes of questions time. So anybody who wants to, feel free to unmute or raise your hand and I'll try and monitor that.
So Merav, I'll start. Wonderful talk as always. This is Jan. Hi there. I know we've discussed this as you alluded to, and I guess what drove me to always think about MCV and the paradox with EPO is a setting of where I started actually first many years ago, which was the human disease of pure red cell aplasia, which is predominantly an immunologic disease.
Yeah.
So if you block with immunologically, so antibody to red cell or T cell, which hits a later stage than you're talking about, because there are plenty of CFUE, at least visually present in the marrow. There's a very high erythropoietin level because there's anemia, and so that's the systemic response. And
Yes.
yet the MCV is sky high.
Yeah.
And so the presumption, just discussion wise, was that if you're driven to make red cells, but you can't because it's blocked at a proerythroblast or just past stage, then the cells continue making hemoglobin and they compensate with a big cell mass. And of course, we've looked at that in terms of heme and not iron. And I wondered what your insights are to that piece of the very old thought process.
Yes, very interesting. So it may be, I guess at the point that the cells are blocked and are presumably attacked immunologically, from that point on, we can't comment. Oh, do any of the cells differentiate into red cells, or are all the circulating cells immature erythroblasts?
No, they're few, so there's anemia, and all the ones that are there are uniformly large.
But they are reticulocytes, they've gone all the way.
They've gone all the way.
Yeah.
It's a small number of reticulocytes because few are being made, and the
Yeah.
cells that exist are large.
Yeah. So what we definitely see, I mean, I haven't been able to talk about it here, but we definitely see that high levels of EPO drive an increase in protein synthesis and an increase in ribosome biogenesis and probably other metabolic pathways. So at least part of the question has to be that they're driving, that it's aporeceptor signaling that's driving the larger cell. But it doesn't, it's still the same answer that you've just provided, because it also drives the synthesis of more globin and therefore more hemoglobin and more heme, I guess. So it's all of that together. So the question might be, why is it that it's advantageous to have a larger cell in the context of anemia? So I have looked into that a little bit because I was puzzled. So in iron deficiency, you actually have a smaller cell, right, in the context of anemia. So what is going on? If you don't have HRI, the cells are not smaller. And also, at least in the mice where this was done, the cells are not smaller and they're not hypochromic. When we looked at the human volunteers, not only was MCV higher, but actually MCHC was lower. And so there wasn't more hemoglobin in the cell. It was that the hemoglobin concentration was lower. So that's the commonality between iron deficiency and high EPO, that in both cases we have hypochromic cells. And maybe what matters is hypochromia. So the way to achieve a hypochromic cell might be to make it a cell that has normal levels of hemoglobin bigger. And all studies that looked at how hemoglobin concentration affects the loading and unloading of oxygen suggest that at lower concentration of hemoglobin, the loading and unloading is more efficient. So I might be slightly misquoting this, but I think what was more efficient was the effect of 2,3-DPG, that was more effective. That's induced in stress. So that effect was better in lower concentrations of hemoglobin. These are cell-free studies, and I don't know if they would hold in the context of a cell, and this is totally hand-waving and speculative. But there you go.
Okay, Mark Perfetto has a question. Mark, do you want to ask your question?
I can. It's a little noisy here. Uh, but
That's fine.
okay, great. Thank you, Dr. Sokolovsky. That was a great talk. It was wonderful. I was wondering if you had looked into iron metabolism genes in your single cell RNA-seq model, like ferritin and PCPB1 and midofferrin and that kind of thing, and whether they get turned on in the early erythropoiesis to ETD switch.
So I I don't recall exactly, but maybe I should. I mean, I think they all do, but I don't want to say every single one, and I don't recall exactly. So I think the best thing is we have maybe I can put a link in the chat. I don't know if I can do that. Um, Maybe Diane, could I send you a link afterwards in an email and you could forward it to people? Because we have all of the genes that we've been able to detect, we have in a very intuitively easy to investigate website. So you can enter any gene you want, and then you'll see exactly where it's expressed.
That would be
Um,
wonderful, Mirav. Thank you for the offer.
yeah,
Send it to me in an email and I will send it out to the group. Thank you so much.
yeah, of course. Yeah. I mean, I think, yeah, I use that myself 3 or 4 times a week at least. And yeah,
Which means the rest of us
it's
will
meant to
use.
be, it's meant to be used by everyone in the community. Yeah.
That's wonderful. Diane Kraus has a question. Diane, do you want to go ahead?
Yeah, hi. I think you gave great talk, CDK2 inhibitor systemically,
Yes.
and I wanted to know if you'd looked at effects on other hematopoietic lineages and then the HSPC, because you're affecting the cell cycle everywhere.
Yes. We have got the data and we haven't paid attention. So I'm sorry, Diane. We
All
should do
right,
it.
so offline, I'd like to know what happened,
Absolutely. Yeah. Yeah.
because a
Absolutely.
lot of fate specification is affected by cell cycles. So
Of course, the prediction based on your work is that there'd be
yeah,
more megakaryopoiesis, of course.
yeah, but
Yeah.
but also I think in granulocytopoiesis, monopoiesis, they're also affected.
Okay. Yeah, no, we should do that. So yeah, the data is all there. Yeah.
Great, thanks.
Okay.
Anybody else have questions? Anybody else just want to say hi? So nice to see everyone. Mirav, that was a wonderful talk.
Thank you. Thank you, everyone.
And I look forward to receiving that link from you, which I will post for
I'll
everyone.
do it right away as soon as we get off.
Yeah, will you unshare for just a minute?
Yeah, so
And I just want to
sorry, that's
remind
easy.
people,
I can put that in the chat too now. Let me try.
that's great. I just want to remind people that CIHD is offering our postdoc exchange again. If you go on our website, you'll be able to see the link And if you have anybody who's interested, let me get there. This one. Okay, can you see that? So this
Yes.
is a description of an opportunity that we have at the University of Utah's Center of Excellence in Hematology. We call it a postdoc student training. It's about almost a week long. It's 4 days where you come out, there's a seminar series, and you get to train at a core of interest that's at the University of Utah. And our cores that are available are the Iron and Heme Core, Protein Metabolite Discovery Core, Mutation Generation Detection Core, and the Metabolomics Lipidomics Core. So if you have a project that's in your lab and a trainee or you trainees who are on might be interested, please reach out to me if you have any questions. This is sort of a description of what you need to send here in the red box. We would love to hear from people and provide this opportunity to labs all over the country to have an opportunity to come and learn about cores at Utah. So I'll stop sharing there and want to thank everybody for their attendance and wave hello. And I'm off to find bears in Yellowstone.
And I just put the link also in the chat.
Perfect. Thank you so much.
All right.
That's a great.
Thank you.
Enjoy.
Thanks, everybody.
I'll do it. Okay, take care, everybody.
See ya.
Bye-bye.
Everybody.